So, a warm welcome to all. Let us begin our new chapter in cytogenetics. In the paper Cell Biology and Molecular Biology, you have to study a module cytogenetics. So, I am going to discuss with the module cytogenetics. The first portion of that module is chromosome bending. You studied the structure of chromosomes, what are the various features of chromosomes, what are nucleosome organization, everything you studied. And also you know some basic idea regarding the chromosomal aberrations, structural and numerical aberrations, etc. So, now we are going to study the technique to study the chromosomal size, shape, the different regions, euchromatin, heterochromatin, the aberrations, etc. we can study by staining a chromosome. The technique is known as chromosome bending. So, what is meant by chromosome bending? So, you know the structure of a chromosome having a centromere and arms, one is P arm and other one is Q arm, that all you know. So, here what you mean by a band? So, a band is actually a part of the chromosome and that is distinguishable from other part or other segments by appearing darker or lighter. Okay. A part of the chromosome, see I will draw you the diagram here. You can have the example, see if this is a chromosome and this is a centromere. So, you can see there are some portions that appear darker, some portions that appear lighter, and this part can be distinguished from other parts that are known as bands. Okay. So, this is a darker band and this is a lighter band. So, the part that can be distinguishable from adjacent segments by appearing darker or lighter is known as what? Banding. Okay. And the technique is actually the method to find out the chromosomal anomaly, that some abnormalities, etc. And this is possible by staining a chromosome with a particular dye. Okay. So, before that, in order to stain a chromosome, before that we have to culture the cell and have to undergo some methodology. After that we will stain a chromosome. Okay. So, let us look at the history of this chromosome bending. In 1960s, Casperson postulated that there is a difference in DNA based composition, difference in the DNA based composition that may produce because of differential intensity pattern along the length of chromosome. Okay. When a fluorescent binding dye is applied to chromosome. So, when you apply a fluorescent means that is fluorochromes, you are apply this dye that will give you the color when you are exposed to a fluorescent light. Okay. So, fluorescent dyes when you apply and you observe, you can see some distinguishable part or there exist certain variations in the intensity patterns in the chromosome. So, this is actually the basis for chromosome bending. Okay. So, from here the concept of chromosome bending was born. Clear? So, that is by differential dyeing and observe under fluorescent microscopy, you can see variations in the intensity of or patterns, and that is a basis of chromosomal bending. Okay. And another one is Buzzkolder and Weaver 1977 also reported differences in binding of non-histones to DNA in different segments of metaphase chromosomes that is involved in the mechanism of G and C binding. Different type of binding is there. It is 1977, it is described by Buzzkolder and Weaver, and 1972 from the Paris conference actually the concept also is derived and many scientists tried to practice this, and finally different types of banding came into practice. Okay. So, why banding? Why we are doing this banding technique? You know, to see smaller pieces of chromosomes and so that we can identify the small structural chromosome abnormalities not visible into routine analysis. So, in usual analysis we cannot identify such abnormalities, but when you do binding, the spelling wrong, when you do binding you can see when you do this binding with the stain or dyes, and you will get chromosome bands, and in that banding you can easily identify the small abnormalities. Okay. So, the stain usually bind with the chromosomes fastly, some specific regions, and you can observe the abnormalities. So, there are two main groups of banding techniques. So, banding techniques, they are mainly based on the region specific or all the chromosomes, all on based on the stain applied. Okay. So, there are two categories, bands distributed along the length of whole chromosome. You can see the bands along the length of whole chromosome, or you can see you can stain only a restricted number of specific bands, that is only a special regions. Okay. That is second category. So, in the first category there are G banding, Q banding, R banding. Okay. These are the most common banding, and in the second case there are C banding, that is centromere specific, and N banding, that is nucleolar organizer region. You studied that. Nucleolar organizer region, the binding or banding techniques means it will stain or banding observe only in that regions. So, that is specific regions. First one is whole chromosome. That is two major techniques. Okay. So, most commonly using techniques are G banding, Q banding, and R banding, and also C banding. Then T banding is also available, that is also specific to the telomere regions of the chromosome. Okay. So, we can look at a glance what are these type of banding. First one is Q banding. Here using stain is quinacridine mustard, that is a stain applied, quinacridine mustard. G banding we use the stain Giemsa stain. So, Giemsa stain is applied in Q banding technique, this is the most commonly used one. And C banding technique again using Giemsa stain, but there is a pre-treatment also applied, and this will stain only the centromere, kinetochore and constitutive heterochromatin. And in the R banding, this is the reverse of G banding or Q banding. G banding reverse. In the G banding, when darker regions are observed in the adenine thymine rich places, but in R banding darker regions are available in the guanine and cytosine rich. So, what is available in G banding, just to reverse that is the R banding technique. Okay. So, in the treatment there are some differences between G banding and R banding. Then T banding, it is a telomere region banding, and N banding nuclear organizer region. Then one more F banding, recently discussing one, Fulgen stain using nuclear organizer region. Okay. So, now let us move on to one by one. So, what happens when you look into this picture, you can see this is a standard staining technique. When you stain, you will observe like this, but when you stain with the Giemsa, you will get like this. Okay. And in the Q banding technique, chromosomes are stained with the quinacridine, and in G banding it is by Giemsa, and the C banding first treated with acid and base, then stained with the Giemsa stain. Okay. So, before going into the chromosome banding, how you will prepare a chromosome or a cell for banding techniques? Okay. For that, first you have to culture the cell, cell culture. Culture the cell, then for up to a 48 hours is enough. Then in that you have to arrest the chromosome division up to the in the stage of metaphase. Metaphase chromosomes are the most condensed chromosomes. So, we can study the morphology and everything when it is in the metaphase chromosome. So, for chromosome banding also we need a metaphase chromosome, clear. So, in the cell culture you will arrest the cell division, that is from metaphase it move on to anaphase. Before that you have to arrest the cell division by applying colchicine, a chemical colchicine. Okay. So, colchicine treatment that will prevent the movement of microtubules and polymerization, depolymerization phenomena of tubulins. So, you will arrest this, and now you have to take that means that arrested metaphase chromosomes, then you have to treat it. Okay. So, usually first treatment is colchicine, then put in a hypotonic solution, then you have to fix it. So, the fixation is usually done in the surface of a chilled slide. Okay, you have to take a chilled slide, then drops of the cell culture placed in that slides, then you can apply all the treatment procedures, especially if you are going on Q banding, you have to treat it with 90%, 70%, 50%, a series of decreasing concentration of alcohol, then phosphate buffer, then stain, that is GIMSA or quinacridine mustard, then you have to look into again into the fluorescent microscope. This is a common technique. Okay. So, a banding, the procedure preparation of method, slide is important. So, first you have to arrest the chromosomal spindle, that division movement, then keep it in the metaphase stage, then transfer the cell culture 1 or 2 drops in a chilled slide, then do the treatments. Okay. So, this is very important, the preliminary step for chromosome banding. Understood? Okay. Now, first we can discuss the first very important type, that is G banding. So, what are the methodology of G banding? So, G banding techniques is also known as that is GIMSA stain, or it by using GIMSA stain, or it is also known as GTG banding. It is also known as GTG banding. Okay. And here what we are doing is controlled digestion of chromosome with the trypsin. That is very important. A pre-treatment with a proteolytic enzyme, trypsin is required here. Pre-treatment with a proteolytic enzyme, trypsin is required here. You know, the nucleosome model you have studied. Okay. So, they are altogether bind with the histones and non-histone proteins. You studied H2A, H2B, all these components, proteins, histone proteins are bind together, linker DNA, everything you studied, nucleosome model. So, here we are going to break up that histone proteins. So, that is done with by using a trypsin, a proteolytic enzyme. So, digest first. But this digestion may vary in the heterochromatin and euchromatic regions. Okay. So, the intensity of digestion may vary in the heterochromatic and euchromatic region. Now, from the culture, from the culture you are taking the cell drops. Culture is actually arrested by using colchicine. Then from that take the drops in a chilled slide, then apply the trypsin pre-treatment. Then we are going to fix this into the slide and applying our stain. Our stain is GIMSA stain. Then you will observe under fluorescent microscopy, you will see the bands. Clear? So, the banding is like this, the alternating bands, light bands and dark bands. Very important point is, dark bands are rich in adenine thymine, that is AT rich. Dark bands are AT rich and light bands are GC rich. Dark bands AT rich and light bands GC rich. This you have to remember because in the R banding just reverse of this process is taking place. Okay. And banding pattern allows the chromosome identification. You can identify chromosomes by looking into this banding pattern. See, this is an image for G-banded metaphase chromosome. See what is happened here, that black color region, that is dark bands, these are the light blue color regions, etc. you can observe here. So, every chromosome, and this is where karyotype is preparing diagrammatic representation. Very simple to identify any of the morphological abnormalities, you can easily identify. And there are bright field G bands, they take their name from GIMSA dye because when you apply this, darker regions that is AT rich tend to be more heterochromatic and they are late replicating and AT rich. And the bright regions that are tend to be euchromatic. Bright regions means our light bands that are early replicating GC rich. So, bright field R bands, in the case of R banding, reverse process, you can see reverse process, dark regions are euchromatic. So, in G banding, dark regions are AT rich, but here in the dark regions are GC rich euchromatic, okay, and bright regions are heterochromatic. Just a reverse of G band, that is the R banding. In the treatment there are some differences, then you will get R bands. Okay. So, just observe the technique. First, arrested chromosomes in a metaphase condition, we apply trypsin or you can apply urea or protease, all are proteolytic enzymes, that is mainly to denature protein, I already told you histone proteins. Then you apply with the GIMSA stain. You can see in the GIMSA stain what happens is that DNA is interact with thiosin and eosin, that are the components present in the GIMSA, that two components thiosin and eosin, that will bind with the DNA, interact with the DNA. then methylene azure and methylene violet or methylene blue or eosin all are present here in the stain. Then you will observe this differential region in the heterochromatic and euchromatic region. One is darker region AT rich and lighter region GC rich. Okay, I hope you understood the G banding technique. Then what are the advantages of G banding? It is used in the identification of band with rich in sulfur content. Okay, so we can identify a band rich in sulfur content, and also identification of chromosomal abnormality that I already told you. It helps to allow you in gene mapping, and also it reflects stronger chromatin condensation. Chromatin condensation, that is the heterochromatic regions you can identify easily. Okay, what are the disadvantages? Mainly one important disadvantage is it is not suitable for plant chromosome because it requires pre-treatment by salt or proteolytic enzyme. Why it is not used in plants? Because plant mitotic metaphase chromosomes are 10 times more shorter than human chromosomes. Clear? So difficult to demonstrate the arrangement of bands at this level. So the size is very small compared to the human chromosome. So we cannot apply this its use in plant chromosomes is very less. So what is G banding, how it is doing, and what are the advantages and disadvantages. Okay. So now let us move on to the second type, that is Q banding, another very important technique of banding, that is Q banding. So in the Q banding technique, again same procedure. From the cell culture, you arrested the chromosome at metaphase in the maximum condensed stage by using colchicine treatment. Then you transfer drops of the cell culture in the slide, and first you have to treat it with alcohol. Is very important treatment. Alcohol that is different percentages, 90%, 70% and 50%. Then you apply, wash with phosphate buffer, that is also important. Then stain with 5% of quinacridate mustard. Clear? So alcohol treatment, buffer treatment, then 5% quinacridate mustard. Then again wash with the phosphate buffer, and then observe under fluorescent microscopy or observed under UV light. Okay. So this is the Q banding technique. Here in Q banding, no pre-treatment is required. You know, in the G banding, before staining, you treated it with the trypsin proteolytic enzyme, but here no pre-treatment is required. That's a very important and very easier method in Q banding. So you will get two types of bands in the banding pattern. One is rich in AT bases and another one is rich in GC bases. Darker regions are AT and light regions are GC, and the darker AT regions, it will dye will quenches, means dye will absorb more in the adenine thymine regions, and you will get the darker bands, and in the lighter bands it do not fluorescence as much that of heterochromatic regions. So heterochromatic regions will absorb more, that is darker than the euchromatic regions. Okay. So that's all about Q banding. So differentiate between G banding and Q banding techniques. See, stained with quinacridate mustard, UV light banding pattern, and both the types, light staining and dark staining. Clear? Okay. Now advantages. It's a simple and versatile technique, and very easily you can apply in all types of chromosomes. You can use Q banding, and this is applicable in the case where G banding is not accepted. And also you can use this technique to study the chromosome heteromorphism and also identify human and mice chromosome. Okay. Then what are the disadvantages? So there is a tendency to fade during examination. The stain will fade while you observe under fluorescent microscopy. So that is the main disadvantage. That is known as photodegradation. Means in the presence of light, it will start to fade. Absorb light of a particular wavelength due to the chemical bond formed between dye and light. That is photodegradation. Then UV light that breaks the chemical bond. That's why that's a one very important disadvantage. Means some regions it's not clear, it will appear like a fade when you continuously looking under the fluorescent microscope. That is about a Q banding. Now let us move on to the next type, that is C banding technique. What is C banding? This is actually banding in the specific regions, especially in the constitutive heterochromatic. Okay. And banding here is caused by differential staining reactions of DNA of heterochromatin and euchromatin. You know, it is more concentrated in the constitutive heterochromatin. So, only specific regions will bind with the stain, and heterochromatic region is usually located near the centromere, you know that. It is usually located in the centromere or in the telomeres or in the nucleolar organizer region. So, specific regions it will selectively bind, okay. And these regions, you know, they are containing highly repetitive DNA. So, those regions that contain the constitutive heterochromatin will stain by using C-banding technique. And this method is a complex technique where we need denaturation and renaturation of DNA, okay. Simply you can't stain, but there are some pre-steps, denaturation and renaturation of DNA. Then you apply the stain. So, DNA denaturation occur treatment with acid. First you treat with acid or alkali or increased temperature. You have to break up our bonds and everything, denaturation. Then subsequently renaturation occurs in treatment with the sodium citrate at 60 degrees centigrade. So, first denaturation by acid, alkali or temperature treatment, then renaturation with the sodium citrate at 60 degrees centigrade. So, this is very important procedure in C-banding technique. Then the repeated DNA or heterochromatin denaturate at low, but that is the high repetitive DNA, but low repetitive DNA and unique DNA do not renature. Only the constitutive high repeated DNA only denature, but other one never going to their procedure. So, there exists a differential staining technique. So, when you apply denature technique, first half, that is those with the highly repetitive part will denature, other nothing happen to other regions. So, you will get a differential staining, some regions only, that is with the constitutive heterochromatin only with the darker bands, okay. So, C-banding is classified by in barley, experiments in barley. Lindlorsen classified this into centromeric bands, intercalary bands, telomeric bands and bands beside the secondary constraints or means secondary constrictions in the short arm of satellite chromosome. These are the 4 visualized C-banding regions according to Lindlorsen in his study in barley chromosome. So, while looking into all these parts, you can see these are the regions where repetitive DNA are located. See, chromosomes by denaturation in alkali solution or acid solution, then by apply sodium citrate for renaturation. So, unique DNA do not renature, only repetitive DNA renature, that is under 60 degrees centigrade for 30 minutes. Then staining with our stain genes, that is G-banding technique is applied here, then banding pattern only in the heterochromatic region. This is a technique applied in C-banding. So, the pre-treatment of the 2 technique is very important, denaturation and renaturation, and apply only in the constitutive heterochromatic region, selective or specific regions only, and last step is by applying with the Giemsa staining. Clear? Okay. Advantages is identification of chromosomes usually in insects and plants, okay, and identification of bivalence at diakinesis. Diakinesis stage, you know, this is the prophase 1 of meiosis 1. So, that you can visible in the centromere region how it is organized. Gene mapping, we can apply this, we can use this study for gene mapping. And disadvantages is it do not permit identification of every chromosome in the somatic cell complement, because you know it selectively bind to the heterochromatic regions, constitutive heterochromatic. So, you cannot get all the chromosomes in somatic cell composition. That is only one disadvantage. Anyway, this is an important technique applied in cytogenetic studies. Now, T-banding, no more details regarding, only it is known as a terminal banding, that is telomere region. Usually high temperature applying and Giemsa staining is applied at high temperature, and banding pattern is efficient for chromosome terminal banding, that is telomere region, and help in identification of translocation. You know the translocation, terminal translocation process. In order to study that, it is very easy. Then end banding, that is nucleolar organizer region. So, first chromosome is air dried, then 5% of trichloroacetic acid is applied at a 95 degree for 30 minutes. Trichloroacetic acid 95 degree for 30 minutes applied and treated with the HCl. 0.1 normal HCl, again 60 degree for 30 minutes. So, this is a pre-treatment. Then again you can apply the gene sustain. Banding pattern is applied only chromosomes that are attached or near the nucleolar organizer region can be visible, and this is in structural non-histone protein linked to NOR region. So, this is very important, nucleolar organizer region, that part only visible by using this N-banding technique. Here pre-treatment trichloroacetic acid and also HCl is applied here. It is very important, specific regions can be exposed. The advantage is identification of both nucleolar organizer region and superior banding pattern, and disadvantage is time consuming technique, and for reagent preparation and everything it is difficult in N-banding. Okay. Then Q-banding was worked out by, so we can see the scientists work behind all this banding technique. Q-banding by Kasperson, I already told you, 1958 it was worked out. Then Jimsa by Summer in 1970, not only Summer, his colleagues also, 1971. And N or NOR banding worked out by Mastui and Sasaki in 1973. And C banding, that is endomeric constitutive heterochromatin banding, was worked out by Linde and Lawson in 1978. Okay. These are the 4 important techniques for you applying. And C banding again Jimsa staining applied, T banding again Jimsa staining applied, and they are all somewhat staining technique is same, but pre-treatment is required. Okay. So, these are the 4 important techniques. Usually the questions from chromosome banding is asking for examinations, so you have to study it. Okay. Now, let us move on to representation of a chromosome band. So, naming or nomenclature of chromosome banding is very important. Okay. So, while going to study in cytogenetics, after banding technique you will get a pattern. So, you have to express it in the form of a formula. So, each common chromosome having a short arm known as P and long arm known as Q, that we are usually given that name, short arm P and long arm Q. Okay. Then each arm is again divided into regions or bands, and that can be observed by proper staining. Okay. Then these bands are labeled. If the bands are in the P arm, you can label it as P1, P2, if it is P2, P3, P4, etc. If it is in the Q arm, you can label it as Q1, Q2, Q3, etc. Counting from centromere towards the telomere. Okay. So, count from centromere to telomere. Now, or at a higher resolution, you can observe sub-bands. Okay. Sub-bands also observed within the bands. That is represented as, if it is P1 band, its sub-band is P11. Okay. Q1 band, its sub-band is Q11. Okay. Q2 band likewise, Q1 band, its sub-band Q11, and another sub-band Q12, Q13, etc. Then sub-bands also available, that you can write Q11.1, Q11.2, Q11.3 likewise. So, a chromosome having P arm, Q arm, P arm having bands, that is band 1, 2, 3, etc. This band 1 again having sub-band, that again having sub-band likewise. So, when you observe under high resolution power, you can observe sub-bands and sub-bands. So, how you will write it? See, sub-bands are numbered from centromere out to telomere. Okay. See, look into this picture, you can see this is a centromere region and this is the P arm, this is the Q arm. Okay. And there are G bands and R bands marked here, and see here how it is labeled. This is a chromosome P arm, this is a Q arm, long arm is Q, short arm is P. Okay. Then see, this is that band chromosome number 17, chromosome number is 17, and its band is chromosome number 17, right first, 17Q, Q is the long arm, 17Q. You can see the 17th chromosome, it's a Q arm we are explaining, and 21, what is 21? Band 21, see band 21, band 23, band 25 here. So, how you will write here? 17, chromosome number 17 on the P arm, band number 12, see likewise. So, first write the chromosome number, then you can write the arm, then you can write the band number, then again you can write the sub band, then you can write the sub band likewise. Okay. So, you can explain this by an example 7q31.2. Okay. What is this? This indicates the gene is on chromosome number 7, that is on Q arm, band number 3, sub band 1 and sub band 2. Understood? Okay. So, the ends of the chromosomes are labeled as PTL and QTL. What is this? Short arm P, it is a telomere TEL, long arm Q, telomere TEL. That is end is marked like this, PTL and QTL. Okay. The 7QTL rougher means chromosome number 7, it is a long arm telomere. Okay. So, long arm of chromosome 7, end of the or telomere of long arm of chromosome 7 likewise. So, remember this terminology, clear. See, once more we can look into this picture. See, this is the chromosome P arm and Q arm. See what is here, marked as bands, light bands, dark bands, light band, dark bands. This is represented as band 1, 2, 3, 4, 5 here marked as, here also it is. See how this is written like this. See what is this? This is actually, this is the petal, means I already told you telomere of short arm. Okay. Then what is this region? This region is chromosome is called 3p21. What is 3p21? 3p21, third band of short arm of 21st chromosome. Clear? Now that we are discussing, and it has subbands. Subbands are 3p22. Subbands are 3p22. 2, 2. Okay. 2 is the band and this 2 is the subband. And again it is divided, subband. So, what you will write? 3p 2, then again 2, then 2, 1. Clear? 3p 2, 2, 1. 2, 1 are the 1 is the subband. If it is right, if you are marking here, how you will write? 3p 2, 2, 2. You will write here 2. Okay. So, if you are marking this, see how it will go? Chromosome number 3. See, that is the first 3. P arm, that is the second P. Then what is the band number 2? See, 2. It's a subband again 2. Subband again 2. It's subband 1, or here it's subband 2. So, what how you will write here? It is 3, then write P, then you will write 2, then again you will write 2, that is subband, then put a dot and write 2. See, this is how you can mark this. From this region also you can mark like that. Okay, clear. So, this is another one more example, chromosome location of a gene. Example, the gene CFTR gene. Where the CFTR gene is located? Look, 7th, chromosome number 7, clear. Q arm, long arm Q, clear. Band number 3, clear. This is the region 3, band number 3, clear. Then band 1, okay, and subband 2. Understood? Okay. So, application of chromosome banding. So, you studied how chromosome banding is done, what are the types of banding, what are the advantages and disadvantages each one, and why, how we nomenclature banding. So, applications, it can be easily applied for identification of chromosome. You can identify any structural abnormality like deletion, duplication, inversion, everything, and also chromosome of cultured cells. You can study by using cultured cells, chromosome banding and cancer. You can study the various diseases, causes, everything. Then C band usually used for paternity testing and gene mapping. So, all these are advantages, and disadvantages is ineffectiveness in certain cases, especially in the translocations, and also failure in detecting micro deletions, okay, and characterizing chromosomes of cell lines which are complex. Such cases we can not characterize the cell lines. So, these are the applications and of chromosome banding and its disadvantage. So, I hope you understood the class and study well. There are chances for asking these questions for 5 marks also and 3 marks also. So, study well and have a nice day. Thank you.